Hi, this is Miss Litton, and this is 7th period review for Honors Bio for Unit 3. Okay, so what are the big chapters that we have here in Unit 3?
Cellular respiration.
Okay, second one, photosynthesis is the third one. What was the first one though?
6.
Yeah, what was 6 about?
That's a good question.
Energy and enzymes?
Yeah.
Oh, yeah. All right. So here's a little list. I've already put this in the stream in Google Classroom. So it's there if you want to use it from there. So here's chapter 6. Here are like 4 big things we can review. Okay? And I'll go over those with you. Here, chapters 7 and 8 are similar. And this is why. Okay? Look what you do. Write the equation for cellular respiration. Identify what gets oxidized and reduced. Then name each major step where it occurs and its purpose. And then you need to do the same thing with what? Photosynthesis. Right? We're halfway through that with photosynthesis. And then photorespiration, you're going to learn about that tomorrow. And then overall, do you know your additional objectives? Then you want to always be able to compare and contrast cellular respiration and photosynthesis. Possibly you make a Venn diagram and you say, how is cellular respiration, photosynthesis similar? How are they different? That would show a really good understanding of those. So my goal with you today is to hit the highlights in chapter 6 and 7. And then that way you have all of that done. So the only thing we're reviewing on Thursday, the day before the test, I'll teach you, finish up photosynthesis tomorrow, and you'll have some time to review, I'm sure. And then Thursday, 7th period, we'll review just photosynthesis, and then Friday you should be good to go for a Hyundai, yeah?
Yep.
All right. So let's look at the big ticket items here on energy and enzyme. What are the laws of thermodynamics? So let's see if you could answer that question right now on your paper. Would you know, or on your digital document, a lot of you are typing, in your mind, you know what the laws of thermodynamics are? Can you remember those? I'm going to give you just a second to think about it. If you're at home, you could be thinking about it too. Maybe writing it down on a piece of paper. This isn't the time to do your math homework. Okay, what's the first law of thermodynamics? Energy cannot be created or destroyed. It can just transfer. Good. Second law of thermodynamics says every time it transfers, it's never 100%, you lose some as heat. Perfect. Okay, so the second one is, how do these laws of thermodynamics relate to entropy? The first thing we need to know is, what is entropy? So think that in your head. What is entropy? Type it down if you're on your own paper. Think it out. What is entropy? It measures something. Don't tell me what yet. Think in your head. You ready? Okay, so entropy measures the amount of disorder. Okay, good. And everything is going to greater and greater disorder. Now, how can we say that everything is going to greater and greater disorder when plants are growing? That's very ordered. Trees are growing. People make babies and babies grow. That's not disorder. I'm going to greater disorder because I'm old. But how is it that things can be ordered right now? Can anybody explain that for me?
Energy. Because using energy keeps stuff in order.
And where are we getting our energy from?
ATP.
Yes, and where are we getting that ATP? How do we get the ATP? Tell me.
Cellular
respiration. What do we need in order to do cellular respiration? Oxygen, and what else?
Glucose.
Food. Where are we getting the food from?
Photosynthesis.
Plants doing photosynthesis, autotrophs, right? Where are autotrophs getting their energy from?
The
sun. The sun is going to greater and greater disorder. Plants are using their cast-off energy of disorder, using it to build sugars and build themselves. That makes sense? And then we can use those plants. So we are transferring that energy, right? That's the second law of thermodynamics. Are we ever 100% efficient in transferring our energy?
No.
No. You know for a fact, because when you work out, you get hot, because not all of it is going into making your muscles move. Some of it is going into generating heat, yeah? Right? So when we relate this to the laws of thermodynamics, All the energy that we're going to have in this system has been made, right? It's just breaking down, and we're able to harvest. The sun is breaking down, and we're able to harvest that energy in order to build up our world. But we're still breaking down too on the whole. Yes? Okay. What is ATP? What does it stand for?
Adenine triphosphate.
Adenosine triphosphate. Good. Adenosine triphosphate. And what has more energy, ATP or ADP? ATP. So what we're trying to do is build ATP through cellular respiration. Yes? Okay. And so we're transferring it. Energy cannot be created or destroyed. It can just be transferred. Are we 100% efficient in our transfer when we do cellular respiration? No. How efficient are we? 39%. Not that efficient, right? If that were to get a letter grade, what would it get? Not enough. Okay? But it's better than what kind of respiration? Anaerobic respiration, which only gets you 2. Yeah, you dropped that class. Okay? All right, so it is our way to do work, and I'm going to show you some pictures. I just wanted to see how much you could pull out. What kind of work do we need to do? What kind of work do animals do?
Structural, mechanical, and chemical.
Chemical work and transport work and mechanical work. So we build things anabolically. Things move, right? And then transport work, like across a membrane. Remember, we have channels and what? Carriers. And some of those carriers require ATP. What kind of transport is that? Passive or active? Active. There you go. Okay. So, are we good on number 1? Yes.
It says chemical transport. What was the last one?
Mechanical. Like actin and myosin sliding past one another, something like that in our muscles. And I'll show you pictures to follow that up. I just thought I would ask you all the questions so we could see if we get them all 100%, but try to do it from here first. Okay, are we good on 1? Do you have any worries on 1? What would be a way to see if you have a good understanding of this information? Yes?
Like, or write it down and check it with the notes.
Okay, write it down, check it with the notes. What else?
Do the chapter review the textbook.
Yeah, you read the chapter, review in the textbook. What else?
The quizzes thing.
The step-by-step. I've asked you big ticket review questions in the step-by-step. What else?
I was going to say teach it, but...
Teach it is a great way to learn it. Yes?
In the review on YouTube for chapter 6, there's always 3 Hello Smart questions at the end, so that I write down the answers. And then
Perfect. Hello, Smart. And I haven't heard anybody, I think, say this. There's questions at the back of each chapter with answers at the back of the book. Did you say that?
Did you say that?
You said that. Oh, okay, yeah, good. What else? There's another one you haven't mentioned yet. Yeah.
On the calendar, like under it, if you go down, there's like questions.
There's questions under the calendar, the objectives, and I heard what you said.
The quizzes.
The quizzes is right? Those are all ways to check to see if you understand it. I would do that, right? Because you want to practice when it's safe. So when you practice for me and it counts for a grade, you're feeling, calm, cool, collected, and confident. Yeah? All right. Describe different types of reactions, exergonic and endergonic. Can you think in your mind, what are the difference between those 2 reactions, exergonic and endergonic? Exit, ender, in. So some reactions require the input of energy. What reactions would those be? Endergonic. Some release energy, that would be exergonic if they release it. Now, what happens if you have one that requires energy and one that releases energy, right? What do you call it when you put them together? Coupled reactions. The release of energy by one can be used to build another. For instance, cellular respiration is a catabolic reaction. It releases energy. We're breaking apart glucose. Let's harvest that energy to make some. A lot of it. Exactly. Okay. Photosynthesis is an endergonic reaction. You have to put energy into it. Where do plants get their energy to put into it? The sun. Perfect. Catabolic, anabolic, we did that too. How are you feeling about 2? Okay. Number 3, how do enzymes work and how are they regulated? How do enzymes work and how are they regulated? In your mind, come up with as many key points as you can about enzyme structure and then jot those down. And then come up with a list of all the different ways enzymes can be regulated. when you're at home, this would be a great idea. Just make sure you have a piece of paper out, write or diagram enzyme structure, what's it look like, what are the key areas, and then make a list. How are they regulated? Maybe pause the video while you give yourself a chance to do that. Okay, are you ready? Okay, when I think of enzymes, I think of a really important spot on an enzyme. What's that really important spot?
Active site.
And that's where the
Substrate
bonds. Okay? And when that substrate binds to that enzyme's going to facilitate a reaction. It could be catabolic, which is about breaking it down, or it could be anabolic, it could be building, right? Okay, there's another site on that enzyme, right? So if I drew an enzyme right here, okay. So this site where the substrate binds, okay, that could be right there. Okay, then I heard you. What's another site that we could have? Yeah, we could have an allosteric site. And that allosteric site, you could have something that binds right here, maybe an inhibitor of some sort. And when that binds, I wonder if these stuck together. Don't stick. Oh, poop. Go away. Oh, it didn't. Okay, but when that binds, it could change the active site somewhat. I had to really change it. So now the substrate can no longer bind, right? And when it can't bind, then you can't do that reaction. Does that mean that this thing is a bad thing? No. It's helping to regulate it. What do you call if something binds right here and changes the active site? Non-competitive inhibitor. But if we went back, okay, and we just remove this, and some other substance bound here, what would that be referred to as? A competitive inhibitor, because it's binding to the active site. Does that make sense? So this secondary site right here where this guy fits in, what's that called again? Allosteric site. Good. Okay. What's another way to regulate enzymes? Besides competitive and non-competitive? Temperature, heat, pH. Good. All of those things, the amount of substrate available to it, right? All those things are going to regulate enzyme activity. Perfect. All right. What is the difference any other questions on number 3 you want to talk about? Oh, coenzymes. We didn't mention that. What is the difference between being oxidized and reduced, and how does it happen? So let's make a chart here. Okay. Oxidized, reduced. Okay, see what you can come up with on your own first about differences between being oxidized and reduced. Without me, see what you can come up with. Do this at home too. I know you can do it. You're so smart. Are we ready? Okay, I'm bored, so let's do it. Okay, what are ways to get oxidized?
Losing the
Lose what?
hydrogen.
Lose hydrogen. So conversely, gain hydrogen. Okay? Okay, give me another one. Lose oxygen. So this one must be gain oxygen. Okay, give me another one.
Lose electrons.
Where do you want me to put that? Lose electrons, and this one is gain electrons. Any other thing?
Lose energy.
Where does that go?
Unoxidized there.
Is lose L-O-S-E or L-O-O-S-E?
It would be loose if it was two O's.
Somebody find out how to spell lose. L-O-S-E, right? Okay.
For some reason,
Lose.
it's O-O-S-E. So
And this would be gain energy.
No, lose is two O's.
Big debate, big debate. All right, are we good on that? Yes, yes? All right, let's check. Let's do some questions. Oh, let's not look at my email. Let me find questions. Let us log in today as whatever dragons, metaphorical dragons you have to slay this week. Maybe you have to do 900 minutes of memory work. I don't know. What are the dragons you have to slay this week?
Ooh, I got it.
So funny, my old thing.
It just doesn't.
3 tests on the same day. Mushu dragon.
Something that you have to deal with.
I wouldn't hug him because he's sick.
He's okay now.
He is? He looked so bad. Okay, good.
My brother. That was me. It's okay. we love
Okay,
each
give
other.
it a go.
T-bone.
Do these at home too. Here, I'll show you the questions. Here's question number 1. Commit your answer to paper. That's the best way to do this, to be authentic. Number 2. And number 3. Let's go, peeps. Let's go, peeps. Come on, come on, come on. Yay! Yay! Aww. Okay. This diagram shows a situation there would be low entropy and high potential. Remember when we talked about that when we have everything together, that's low entropy, high potential energy. Yes? As it disperses, entropy increases. All right. All right, let's try again. I'm looking for more questions. Here we go. Try these at home. Are you ready? Here's question number 1. Question number 2. Pause if I'm going too quickly. And here is question number 3. When you listen to these reviews at home, do any of you listen to these reviews at home?
Yeah.
Do you do the questions again?
Yeah.
Does it help? Yeah. I am freezing. Okay, we ready? All right. Aww. All the chemical reactions that occur in a cell is called your metabolism. Enzymes just help you do them. Number 2. Yay! Aww. The energy of an ATP molecule is released, all 3 of these. ATP becomes ADP plus phosphate. ATP is hydrolyzed. ATP is coupled with an endergonic reaction. Okay, let's try another one. Ooh, you know what we didn't talk about? How enzymes lower the energy of activation. We didn't review that in our review. Let's make sure we know that. That's what he's off in class, okay? Enzymes lower the energy of activation and make the reactions more likely to occur. That's why they speed up reactions. We never said that as its purpose. That's kind of key. Okay. Kind of key. Here we go. And here are the questions. Here's one. Remember, pause it if I go too fast. Here is two. Write it down. Don't just think it in your head. Write it down. Here's number 3. I'll make this diagram bigger for you. Watch. Oh, yeah. Look at the diagram because it's going to go away so you can see the question. Stare it down. Okay, good. And here, oh,
my brother knows what Miss Listen's voice sounds like, even though he never met her.
here's question number 4.
It's like funny, like I'll be at home whenever I talk to her, just keep going.
Okay, we're all done. Ready?
No.
It said 100%. Okay. Oh, it was only question number 100%. My bad. I'm sorry. Okay. We've got to do. The active site of a specific enzyme is similar to that of any other enzyme? No. It is unique. Is the part of the enzyme where the substrate can fit? Yes. Can be used over and over again? Yes. It is affected by temperature and pH. It is affected.
Oh, I didn't even do that one.
Okay, number 2. If you want to increase the amount of product per unit time of an enzymatic reaction, do not increase. So all of these will increase the amount of product. The amount of substrate will increase it. The amount of enzyme, increase the amount of enzyme, you'll have more product. Increase the temperature a little bit, not so much that it denatures, but a little bit, that'll work. But if you change the pH, remember every change in pH is a tenfold change. Good. Number 3, this diagram. Yay! Those that answered it, yay. Aww. The allosteric site is the same as the active site? No, it's not the same. Non-protein, no, it's still protein. No, it often involves feedback inhibition. We just went over that. Okay, and let's see, is this? Oh, I guess there are no more. Yay! You can do you can do the dragon.
Oh, yeah, do dragon.
Do the dragon. Since we're slaying dragons, do the dragon.
Wow. I know, and you're like, no, I don't want to eat that. I want to eat that.
First. No, I was first. I said first before. I didn't get any.
Well, that was a mistake.
Lose electrons. Where does lose electrons go?
Oxidized.
Gain hydrogen. Where does that go?
I got the oxygen.
Gain electrons. Where does that go?
Reduce.
Lose oxygen. Where does that go?
Reduce.
Lose hydrogen.
Oxidized.
Lose energy.
Oxidized.
Yay! Dragon and I are friends. All right, perfect. Okay, now back here.
Oh, I'll send you a video.
Okay, now, next part. Basically, it is write the equation for cellular respiration and photosynthesis. You could do them both at the same time. Identify what gets oxidized and reduced, and then name each major step and where it occurs and its purpose. All right, so if we were going to write that equation, so I could write it both ways, I'm going to put energy here. Okay, and if I did energy plus 6CO2, what else do I need to say?
Plus
Oh, my goodness.
C6H2. Oh, wait, no.
Goes to what?
C6H2.
Oh my gosh, it's so messy.
Plus
Oh wait, mine was wrong.
6O2. And if I go the other way, then I'm doing what? Yeah, this is photosynthesis. And this is cellular respiration, right? And the energy here would be from the sun. And when I go this other way, it would be what? ATP. All right. Glucose becomes CO2. Oxygen becomes water here. And
Oh my gosh, I did the most amazing flowchart.
okay, CO2 gets what into glucose? What happens to CO2? Reduced. And the water gets oxidized. Okay, and in cellular respiration, the glucose gets what?
Oxidized. I said oxidized.
And then the oxygen gets reduced. Okay, this occurs in the mitochondria. Could you name all the places? And this occurs in the and could you name all those structures? Yep. What's the name of the energy transfer molecule?
No, that's the member.
Oh, an NAD.
Yeah. Yeah? Got it? Okay, there you go. Just waiting for the paparazzi.
What's the energy transfer? Wait,
Anything else?
oh, no. I thought you were talking about the group.
And then you could name all those structures, right?
Not for chloroplast.
Okay. Can I move off this? Are you good? All right. And then here it says, write the equation, identify what gets oxidized, reduced. Did we do that? Now we just need to name each major step. So I can think of no better way to do that than to sing. Okay, so let's do cellular respiration. What's the whole thing called?
Cellular respiration.
First step?
Glycolysis.
Who says it?
All cells.
Where?
In the
What
cytoplasm.
do we start out with?
Glucose.
How many carbons?
6.
What are we going to do?
Break it in half.
Step?
10.
Enzyme?
10.
Called?
2 pyruvic acid.
Andrew,
Reduce some NAD and make some ATP. 2.
Oh, wait, we have oxygen. What should we do?
Aerobic respiration.
What's the next step called?
Transition prep.
Here we go. And you
Reduce some NAD and 2 acetyl-CoA put 1 away.
Pause.
What's the next step?
Yeah, but I want you to tell me, glycolysis occurred where?
In the cytoplasm.
Transition prep at where?
In the
The matrix.
Oh, wait, no.
Transition prep is in the
mitochondria. In the matrix.
matrix of the mitochondria. And the next reaction is called the? Where's it going to occur?
In the cristae of the mitochondria.
In the Krebs cycle matrix of the mitochondria. What's the next step after that?
What is my
ETC, where's that going to occur? Cristae of the mitochondria. Review that. Okay, wait a minute. Just wait a minute. Okay. So here's our cell, right? So glycolysis. Okay, I still have my acetyl-CoA here. Prep and Krebs, and then ETC occurs there. Go ahead, I know you want to do it. Just take a picture. Okay.
Oh, wait. Yeah, you're right. I'm sorry, I'm slacking.
Here, wait, let me make this better. Then on those thylakoid membranes, that's where you do the light dependent. And then out here is where you do the light independent.
Wait, so do you have to do both of them or just one of them?
No, you gotta do them both.
Okay.
That's like saying, do you have to do the transition prep and prep? Okay.
Oh, because like.
Do you need me to start the song over from the beginning so you have it? Is that what you were trying to do? Or are we okay at transition prep? Here we go. They're not recording it, they're fine. Transition prep, here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. 2 plus 4 is 6, do the bit, get 5, do the bit, get 4. Make an ATP, reduce an NAD,
Reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs, take it to the top of ETC. Reductase, cytochrome oxidase.
And two makes
Makes an ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it.
Catch the electrons at the bottom.
Oxygen.
Forming
Water.
water. Want to see something? Water. Water,
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 water, water, water.
That's what we do. Because there are 6 waters, right? In the equation. Water, 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 water is 5, but the 6th one,
No way.
water. Way.
stop.
They loop. They loop. Huh? Because in life, they loop. Huh? Okay, and now we need to do, here we go. How many carbons?
6.
What do you need to do?
Break it in half.
Step?
10.
Step? 10. What are these called?
2 pyruvic acid.
And you?
Reduce NAD and make some ATP. 2.
Oh no.
What do you do?
Fermentation.
How many choices do we have?
2.
You and I make
Lactic acid makes our muscles sore.
plants and ants make
Alcohol.
Either
way, you oxidize your NADs.
Okay, so we'll finish up photosynthesis tomorrow, and we'll be golden, Pony Boy. Tomorrow in class.
Oh, in class.
Yes.
Then we have the review.
Yes, and then we have a review. Okay, love you. Make good choices.